Hello, my name is Giselle Piedra, and I am a senior at John Jay College of Criminal Justice pursuing my Bachelor's of Science degree in Cell and Molecular Biology. Today, I will be telling you about the research that I have been doing in the epigenetics lab of Dr. Delgado-Cruzata, where I look at the effects mitomycin C has on the expression levels of microRNA 125A, 5P, and MCF7 breast cancer cells. Mitomycins are highly potent antibiotics that are used as chemotherapeutic agents to treat various forms of cancer, including breast. These compounds are able to alkylate DNA upon reductive activation to generate monoadducts. Single-stranded DNA is more prone to adenine and guanine alkylation than duplex DNA. This suggests that single-stranded nucleic structures such as RNA could be targets for alkylation as well. This was further supported by reports of ribosomal RNA formation of mitomycin adducts in vivo. With this research, we wanted to learn whether mitomycin C affects the levels of microRNAs in breast cancer or whether it interacts with microRNAs in a similar way as it interacts with DNA. We focused on microRNA 125A 5P, which belongs to a type of small non-coding RNA molecules that regulate gene expression by RNA degradation and by inhibiting translation. MicroRNA 125A 5P regulates the expression of proteins known to inhibit cancer cell growth and metastasis. It functions as a tumor suppressor in ovarian and breast cancer. We determined the expression levels of MIR125A, 5P, and MCF7 breast cancer cells that were exposed to 50 micromolars of mitomycin C for 24 hours by extracting total RNA and carrying out reverse transcription and using quantitative PCR to measure MIR125A, 5P levels with RNU6 as the reference gene. We also used prediction software to predict the MIR125A, 5P, 2D and 3D structures and created models that contain the potential mitomycin adducts. We found that treatment with mitomycin C significantly decreased the expression levels in MIR125A, 5P as shown in Figure 5. We predicted 2D secondary structures, which are shown in Figure 6, where we identified 5 guanine and adenine nucleotides that would form adducts after treatment with mitomycin C. We constructed a fragment of MIR125A, 5P, shown in Figure 7, that has 4 nucleotides in order to determine the formation of adducts by alkylation of mitomycin C and any alterations that occur. We were also able to predict the 3D structures of MIR125A, 5P that are shown in Figure 8. With these structures, we were able to identify which regions of MIR125A, 5P will be altered by the formation of adducts. The nucleotides in red represent the guanine, and those in blue represent the adenine. The results from these experiments suggest mitomycin C downregulates MIR125A, 5P in breast cancer cells. This effect might be mediated by altering its 3D structures. Overall, this work suggests that mitomycin C has the potential to target microRNAs and modify its functions by affecting the amount of microRNA present in the cell. In future experiments, we hope to carry out simulations of the modified MIR125A, 5P with the mitomycin C adducts to understand the mechanisms that mediate the disruption of microRNA function. Lastly, I would like to acknowledge the John Jay Office of Student Research and Creativity for supporting my project. I would also like to thank my mentor, Dr. Delgado-Cruzata, and my lab group for their guidance and encouragement. Thank you.